Hello, I'm Bajuna Salehe. I'm from the McGuffin Group at the University of Reading, and I'm founded by the BBSRC. In this presentation, I'm going to talk about IntFOLD, which is a free-to-use server for predicting the 3D structure of proteins and their functions from their amino acid sequences. IntFOLD is a popular web server for predicting 3D structure of proteins and their functions from amino acid sequences which are given as input. IntFOLD has served many users worldwide from over 150 different countries. As of late June this year, the number of unique users is approaching 24,000, and the number of jobs completed by the server is approaching 300,000. These plots show the growth in the number of unique users of IntFOLD and the number of jobs completed over the years it has been active. By looking at these plots, it is clear that there has been continual growth over time, both in the number of unique users and in the number of jobs completed by the server. This growth is expected to continue, and the red lines show the extrapolated growth curve fitted to the blue data points. So why are so many people who want to use the server to predict the structure and functions of their 3D protein targets? Well, the main reason is that there is continually increasing gap between the number of available sequences in UniProt and the number of known structures in the PDB. Even now, with the advent of AlphaFold, which has significantly increased the hope of reducing this gap, there will still be gaps in the data regarding novel sequences, sequences with the disordered regions, protein interactions, and our knowledge of protein ligand binding sites. Additionally, AlphaFold models are often not perfect, and there is still room for refinement and the optimization of these models to truly close this gap. So tools such as IntFOLD will help us to do this and also to further realize the promise of next generation sequencing in genomic research. So why using IntFOLD? Well, IntFOLD has user-friendly interface with a very simple submission system and intuitive output. Apart from predicting tertiary structure, IntFOLD integrates several key aspects of structure and function prediction, including model quality assessment, ligand binding prediction, domain prediction, and disorder predictions. Users can also use IntFOLD to refine their models further. It has leading performance compared with the popular state-of-the-art methods, and I will show some benchmarks on this shortly. So how does it work? Under the hood, IntFOLD incorporates both template-based modeling and free modeling methods using consensus-based approach, and it produces many alternative models for a single target. It provides built-in quality estimates for these models in order to usefully score and rank them. The server also uses the 3D models to help predict the ligand binding sites, domain structures, and the disordered regions within the target proteins. This slide shows graphics representing the unified component methods of IntFOLD. At the core of IntFOLD is ModFOLD, which provides built-in quality estimates of each model. The model shown here is colored by its predicted parity accuracy from blue to red, blue being accurate and red being inaccurate. From this image, it is clear that, or it is easy to tell at a glance which part of the model can be trusted and which part of the model may be inaccurate. Additionally, the server uses IntFOLD to predict structural domains and is able to color these structural domain differences of the model. The server also uses a disorclast method to predict disordered regions of the model. IntFOLD, it is another method integrated in the IntFOLD, which is used to predict the ligand binding site of the protein target. Finally, users also have the option of resubmitting any of the models for further refinement using refold method. This slide shows an example of the accuracy and the completeness of predictions that are obtainable from the IntFOLD server. The 3D model and ligand binding site were accurately predicted for CASP target T0971. This was in CASP 13, by the way. Furthermore, the IntFOLD server correctly predicted the N-terminal unstructured domain, which could not be observed in the crystal structure. Okay, so how do we use IntFOLD in the daily basis, the system? So how do we interact with the system? Performing predictions using IntFOLD is very simple. You just input the amino acid sequence for your target protein in this submission form. So this is an example for amino acid sequence. You may optionally write your target name and email address. Then you click predict button. These are screenshots representing different job statuses which the server provides to user after submitting target. The status include pending, waiting, and running. The queue data is also reported, and once your jobs are completed, you'll see the results page at the link. The following link provides an example of an output page where it shows the top 5 predicted models. The version of the server is participating in the current CASP 15 experiment, and this is IntFOLD version 7, which is also in Cameo, and I'll show this shortly. On this result page, the first table shows the top 5 ranked models with the model ID, confidence value of the model, global model quality score, local model quality plot, which shows the predicted per residue error. You can also click a button to view each model interactively in 3D and download the PDB files. There is also a button to fix any identified errors in your models using the latest refold method. Scrolling down the page brings you to the disorder probability plot and the domain boundary prediction and the IntFOLD protein ligand binding site prediction, which can also be viewed interactively. Finally, the last table shows the full model quality rankings of all generated models. Further improvements to IntFOLD are currently in the works, which are being tested out at CASP 15. These include IntFOLD version 7, which integrates new deep learning methods for modeling tertiary structures. ModelFOLD version 9 for quality estimates, which also integrates several new deep learning methods. IntFOLD version 4, which integrates tools for ligand redocking, MultiFold for modeling quaternary structures, and the ModelFold doc for providing quality estimate for quaternary structure models. Each IntFOLD version has been independently evaluated in terms of its performance. Over the years, it has been ranked among the top performing servers based on the two major independent benchmarks. One benchmark is called CASP, Critical Assessment of Protein Structure Prediction, which is an biannual experiment. And another one is CAMEO, which is Continuous Automated Model Evaluation. In CASP 12 and CASP 13, IntFOLD was one of the top performing servers overall among those that participated in the experiment. But it is also the leading UK-based servers in that experiment, or in those two experiments. In CASP 14, it is also performed relatively well compared with the established groups, but then we know all that in particular year, the AlphaFold 2 performance outclassed all other groups. In this table, the performance of IntFOLD 7 is compared with the previous versions and other popular publicly available servers such as RosettaFold, Swiss Model, and Fire2. The comparison was done on common subsets 3 months worth of CAMEO data. The reference method is IntFOLD version 7, and it is shown to outperform all other methods according to all metrics. In addition, the older version also performed comparatively well versus other leading servers. This plot shows the performance of ModelFold version 9 compared to other leading public servers for model quality estimates according to the CAMEO benchmarks. There are two variants, ModelFold 9 and ModelFold 9 Pure, both of which are outperforming the majority of the other web servers. The ModelFold 9 quality estimates are integrated with IntFOLD version 7. This is how to cite IntFOLD. These are the latest two papers describing the integrated server. There are other papers which describe each of the component tools that have been published more recently. This is the IntFOLD team led by Professor Liam McGuffin, who is a PI in this project. Some of these members are PhD students and others are postdoc researchers. Thank you for your attention.